This video covers the IGCSE Biology Objective 5.1.6 on the 2025 CAIE syllabus to describe the steps of enzyme action. So we've seen this before. Enzymes have active sites depicted here with this arrow. And just as a reminder, the active site of an enzyme is where the chemical reaction actually takes place because the chemicals in question bind to that specific location of the enzyme itself. They bind to this active site. So during the process of enzyme action, we have something called the enzyme substrate complex that forms. It's also called the ES complex. And basically this is when the substrate has joined to the enzyme. Prior to creating the new product, the combination of the enzyme and the substrate together is called the enzyme substrate complex. And so basically this is a temporary molecule that forms when the enzyme comes into perfect contact with the substrate, and that is going to allow the chemical reaction to take place. Now let's not forget that these interactions, these contacts, are due to random movement of the molecules, and we know hopefully what some of the factors are that will allow for this random movement to happen quickly, so the enzyme substrate complexes will continuously form as the enzyme is doing its job. All right, so let's go through enzyme action. First of all, we know that there are enzymes and there are substrates. They have to come into contact with each other. This can be controlled by any number of factors, but we want the substrates to come into contact with the enzyme as quickly as possible. One of the factors that can contribute to this is temperature, because we know that heat energy can convert to kinetic energy. That's going to cause the substrates and the enzymes to move faster in their solution or in their area wherever they are, and so that is going to allow for the possibility of these interactions to occur much more quickly. When we have the substrate or substrates binding to the active site of the enzyme in perfect contact, we then have formed what's called the enzyme substrate complex. And the enzyme does the chemical, then the chemical reaction occurs. Once the chemical reaction has happened and we have a new product formed, then the product is no longer going to stay in the enzyme. The reason why it's removed from the enzyme is because it's not the substrate molecules. It's the substrate molecules that forms the enzyme substrate complex, not the product combined with the enzyme. So once the product has formed, the enzyme no longer recognizes this structure and will release it. So that now allows the enzyme to now go back and find new substrates and repeat that process. So let's just quickly take a look at how this works chemically in a specific molecule. So we have sucrose molecule here. It's going to be hydrolyzed. That means it's going to require a water molecule to split the sucrose molecule into its components, which is glucose and fructose. And that is going to require an enzyme. And you can see that the enzyme has a shape that is complementary to the sucrose molecule. The water molecule comes in to enable the enzyme to split the molecule into glucose and fructose. And then once those two products have formed, the enzyme releases the product in order to allow for a new sucrose molecule to bind to the active site.